After watching this video, you should be able to describe the feedback control of plasma glucose, list the important organs involved, that would be the endocrine pancreas, the liver, skeletal muscle and fat, and the hormones insulin and glucagon. Once you put it all together, you should then be able to apply it to situations like fasting, the well-fed state, even type 1 or type 2 diabetes mellitus. So let's take a look at the endocrine pancreas first and those beta cells. And the beta cells secrete insulin. So insulin is going to be coming out of the beta cell. And insulin has receptors on the liver as well as the skeletal muscle and fat. And the relationship between insulin and these different physiological functions are the following. When insulin goes up, hepatic glucose production goes down. When insulin goes down, hepatic glucose production goes up. So that is a negative relationship, which we will indicate with a negative sign. When insulin levels go up, skeletal muscle and fat insert glucose transporters called GLUT4 into the surface, takes up glucose from the blood into the cells, and glucose goes down. When insulin levels falls, the glucose transporters get sequestered and glucose uptake goes down. So this is going to be a positive relationship. So you can see that insulin's job really is to lower the plasma glucose by suppressing hepatic glucose production and promoting glucose uptake from skeletal muscle and fat. Let me just say that even though we have here for the liver hepatic glucose production, the liver also takes up glucose. So when insulin suppresses hepatic glucose production, it also at the same time really is promoting liver uptake of glucose. Well, we don't have that shown in this diagram. Now let's take a look at the liver. And the liver, like we said, has the ability to produce glucose. It's only one of two organs that can do that. It's the primary organ in the body that does this. The other organ's the kidneys, which has a secondary role in this process. But in this case, when the liver hepatic glucose production goes up, plasma glucose goes up. And when hepatic glucose production goes down, plasma glucose goes down. And that's because the liver is releasing glucose into the blood when it's engaging in hepatic glucose production. And when skeletal muscle and fat are taking up glucose, plasma glucose tends to go down. And when glucose uptake goes down in skeletal muscle and fat, that tends to cause a rise in plasma glucose. Now, if we go and finish off this feedback loop, we have, well, the plasma glucose actually comes around and can have a direct effect on the beta cell to promote insulin secretion. So when glucose levels go up, insulin release goes up, and when glucose levels goes down, insulin release goes down. And there's a very specific mechanism of how that works, which will be shown in another video. So this shows that we have a negative feedback loop. In fact, we have two parallel ones that are working together to stabilize the plasma glucose. We can see that we have one inversion if we follow this loop around, okay, going from insulin to hepatic glucose production to plasma glucose back to insulin. And then we also have another negative feedback loop going from insulin to skeletal muscle and fat glucose uptake to plasma glucose and back to beta cells again. See, we have an odd number of inversions, which indicates negative feedback. Now the other hormone that we can focus on here would be glucagon. And glucagon comes from the alpha cell. And we can show that over here. And in this diagram, glucagon receptors are really only going to be important for the liver, hepatic glucose production, because it turns out that, first of all, skeletal muscle don't have glucagon receptors. Now fat cells do, but it regulates hormone-sensitive lipase and lipolysis, not really glucose uptake. So in this diagram, we're not really going to be having glucagon going over to fat cells. We're just going to have it over here with the liver. And you can see that if we want to finish off this feedback loop, we need to have something connecting the alpha cells to this whole system. Now it turns out that there might be a mild effect of plasma glucose on, go draw it over here. There might be a mild effect of, we can kind of put this as a dotted line to suppress alpha cell release of glucagon. Now the reason why I'm making this dotted, I'm not saying this is the primary mode of glucagon regulation, is that the beta cells get blood flow first, and a lot of the beta cell products that are coming out are going to reach the alpha cell in a very high concentration, and it turns out that insulin actually has a negative impact on the alpha cell. Okay? In fact, the beta cells may even be releasing GABA. They do have the enzyme glutamic acid decarboxylase, or GAD, and it might even be GABA that's inhibiting this as well. Now, this completes our feedback system because if we follow glucagon, we have glucagon going to the liver, to plasma glucose. There's one inversion, so that's negative feedback. And if we follow it all the way around, going through insulin, there's one negative sign too. So we have really multiple parallel negative feedback systems all set up to stabilize the plasma glucose. And the only other thing that I'd add is that the beta cells release in equimolar amounts something called C-peptide. Okay, I'll put that in red. Okay, and the reason why that's good to have out here is that the beta cell release of insulin, which is an endogenous source, always goes along with C-peptide. So if we were to measure C-peptide in the blood, that would tell us about endogenous insulin secretion. You know, so if we measured insulin in someone's blood and it was high, we really wouldn't know if it was someone injecting insulin or it was coming from their own pancreatic beta cell. So we put beta cell, insulin, and C-peptide down here. It's useful to keep that in mind. Notice that I have it in red. There is no arrow going from C-peptide anywhere because C-peptide doesn't really do anything biologically. It's just really a marker tells you about endogenous insulin secretion. So now that we have this whole feedback system set up, what we can do is we can think about, well, what would happen with some different scenarios? Okay, so if we think about, we can start with well-fed state. You go over here. We can let's put eating. Okay, let's say you're eating a bunch of carbohydrates or something. If we think about where that would fit in first, eating really would have an effect on plasma glucose first. And so when we're doing these sorts of feedback loops, we want to know where to start. We're not going to go necessarily left to right. We want to go the logical place where this would be fitting in our feedback system. So plasma glucose should be going up if you're eating a carbohydrate meal, and we can have that indicated with an up arrow. And then we go back to our feedback loop. If we go in and think about how this would all play out, glucose would go up. That would cause insulin secretion and C-peptide both to go up together. And so that would be the next place we'd want to start finishing off this diagram. And so insulin and C-peptide would go up. Now, if insulin goes up, we know glucagon goes down. And if insulin and glucagon are in this sort of profile, we know glucose production by the liver is going to go down. And skeletal muscle and fat are going to be inserting those glucose transporters in the membrane, and glucose uptake is going to go up. So you can see here that we had something that disturbed the plasma glucose and caused it to go up, and now we're having a physiological response of insulin going up and glucagon going down to cause these organs to respond to stabilize the glucose and bring it back down to a normal level. So the glucose wouldn't be up for long. After a meal, it goes back down because of this response. Okay, now let's look at fasting. Put in fasting over here. When you're fasting, the plasma glucose is going to go down. You're not eating, so that would be low. And then if you go over here for insulin and C-peptide, right, that's going to be suppressed. As a consequence of insulin going down, glucagon is going to go up. All right? And if glucagon is high and insulin is low, then you're going to be having your liver glucose production go up, and you're going to sequester those glucose transporters in these tissues. The glucose transporters in skeletal muscle and fat are going to be inside. They're not going to be taking up glucose, and those transporters, by virtue of them not being around, the glucose uptake is going to go down. So this is helpful because it's going to help shunt the glucose to vital organs, like, for example, the heart or the brain, and not have the skeletal muscle and fat taking them up. This would be the appropriate response to a disturbance that's causing a low plasma glucose. Again, these responses are stabilizing the glucose, preventing a further drop, and even trying to bring it back up to normal. So now let's take a look at type 1 diabetes mellitus. So we'll call this type 1 DM for diabetes mellitus. And in type 1 diabetes mellitus, there's an autoimmune destruction of the beta cell. So you're losing beta cells, something like once you get down to maybe 20% or 10%, you have to knock out quite a high percentage of beta cells before you start to show problems, your glucose going up. And so the insulin and C-peptide are going to go down because that's what's being destroyed. So we'd start there. So insulin is down, C-peptide is down, and now because insulin is low, glucagon is going to be high because remember insulin suppresses glucagon, maybe even GABA a little bit as well. And then hepatic glucose production, is going to go up, and those glucose transporters are going to be sequestered, and glucose uptake is going to go down. And your glucose is going to be high. All right. So notice the difference between this scenario and what we were just doing before, right? In eating and fasting, the plasma glucose is the first place that we started, right? Over here, it was the insulin and C-peptide was where the disturbance was. And if you follow the comparison of fasting and type 1 diabetes, let's say, you can see that it's very much the same. Insulin and C-peptide are both low, glucagon is high, hepatic glucose production is high, glucose uptake in skeletal muscle and fat is low, but the main difference here is the glucoses are different, right? So these responses were trying to stabilize the glucose and bring it back to normal. In this case, this plasma glucose is a consequence, a product of this disturbance over here in these hormones. And so if you know what is going on in the fasting state with respect to insulin and C-peptide and glucagon, hepatic glucose production and glucose uptake in skeletal muscle and fat, you really know what's going on in type 1 diabetes, right? The only difference is that the glucoses are different. Type 1 diabetes, the glucose is high, and in fasting, the glucose is low. That's an important thing to consider. Now, if we go and look at type 2 diabetes, it's a lot more complicated. It's a lot more complicated. And what we want to say with type 2 here, if we were going to use this as an example, all right, is that there's an insulin resistance, there's a decreased effects of insulin at the intracellular level, the transduction pathway level. And what initially happens is the plasma glucose is gonna go up because you're having the insulin effects are going to be not working very good, right? So you have glucagon is high, hepatic glucose production is high, skeletal muscle and fat glucose uptake is low, all right? And plasma glucose is elevated. And because plasma glucose stimulates insulin, there's going to be an increase in insulin and C-peptide, okay? And the thing about type 2 that makes it a little tricky is that, if someone has insulin resistance and their beta cells can respond very well and secrete lots of insulin, you might even see, before type 2 even gets diagnosed, you might even see the glucose in the blood be somewhat normal because the beta cells are able to secrete enough insulin and C-peptide. So the way we have this laid out over here, the way you'd think about it is that even though the insulin is elevated, all right, if the plasma glucose is elevated in type 2, this insulin is insufficient to counteract and overcome the insulin resistance that's making it so it can't have its effects. Okay, so I just want to point out that before someone's even diagnosed with type 2 and they just say they have insulin resistance and their glucoses are normal, you would see still a hyperinsulinemia and high C-peptide because that's the appropriate response to the insulin resistance. And it's really only when the beta cells can't do it, can't make enough insulin for that individual patient, that this glucose is elevated. But for the purposes of our feedback system, this fits in perfectly with what we just did when we're working out how that would work, because we're really starting at the insulin effects first and working our way around the loop. So hopefully these examples help solidify the stabilization of plasma glucose mechanisms that are in place in the body normally in response to eating and fasting, and then what happens when you have disturbances in the system. In this case, we looked at type 1 and type 2 diabetes mellitus. And that concludes the video on stabilization of plasma glucose, looking at feedback control.